Hello students, we will be talking today about sterilization or we could say control of microorganisms, because all the methods, all the techniques that we are using is not sterilization. The different terminologies with respect to control of microorganisms we have studied in the previous class. So let us see what are the different methods of sterilization or controlling the types of different methods of controlling microorganisms. So if we have to talk about definition, it's making a substance free of microorganisms, may it be vegetative cells or may it be spores or may it be any kind of microorganisms. It should be free. There has to be zero microorganisms here. So that is called as sterilization. Okay, there could be other methods also wherein you will reduce the number or you will stop the growth, but then sterilization is not any of that. Sterilization is completely making the substance free of microorganisms. So there are different types of sterilization techniques, or there are different types as to how you could control the microorganisms. One is physical methods, chemical methods. In physical methods, you have sun drying, filtration, dry heat, moist heat sterilization. Chemical methods, there are different types like liquids, solids, gases, and there is another group or another category which is called as radiation, that also could come under physical methods, and ultrasonication. About all these techniques we would be studying in detail in the future slides. Before that, we need to just know something about TDP and TDT, thermal death time and thermal death point. What is thermal death time? It is the time required to kill the microorganism at a specific temperature. That is called as thermal death time. How long a microorganism takes to die at a specific temperature and a specific time. So that is called as thermal death time. What is thermal death point? The least temperature to kill the microorganism, one specific microorganism, at 10 minutes. That is called as thermal death point. Moving further, the first type, I had spoken to you about the physical methods, that is sunlight, drying, filtration, dry heat and moist heat. Talking about the first type here, sunlight. We use different methods to control the microorganisms. One of the method is sunlight. We know that sunlight has UVs, ultraviolet radiations, and through ultraviolet radiation, microorganisms are destroyed through mutations. Okay, there are certain changes that happens in the DNA or the genetic material of the microorganism. Thus, it will modify or destroy the microorganism. So, basically, this is not a sterilization technique, but it is a disinfection technique, because it will reduce the number of organisms. Okay, here the penetration power is very less. The UV penetration power is less here. Okay, so it will not penetrate into a substance, but only superficially the organisms will be reduced or killed here. Okay, generally we can see this happens in water. You might have seen natural waters. They are supposed to be clean. They are supposed to be having less load of microorganisms. Why? Because they are continuously exposed to sunlight. The river water, the sea water, they are continuously exposed to sunlight. So thus the load is very less there. Okay. Not only that, you might have also seen in your houses wherein you keep your food materials for under the sunlight. When you do that, definitely the shelf life of that particular food or that particular substance increases as the number of microorganisms becomes less there. Drying also, you could say, one method of putting it under sunlight, but you can also apply drying by other physical methods too. Drying is elimination of the major part of the water content of that particular substance. By drying, you would not allow the microorganisms to proliferate, to increase in number, to multiply, to carry out their metabolic activities. If you see, water, 80% of the entire cell's body is made up of water, and water is definitely required for the microorganisms to carry out their metabolic activities, to carry out their various activities. Now, if you restrict water, definitely organisms cannot multiply in numbers, so you are stagnating their growth. Example, like your pulses, your dal, etc. So these materials, dal or the dry rice, which is in the dried form, you can store it for a very long time, but the same rice, when cooked, You cannot store it for a long time because it is having some amount of moisture. The same soaked dal or soaked rice, you cannot keep it for a very long time because it is kept for a long time, and because it is having moisture in that. The shelf life of the food will increase in the dry state because the number of microorganisms are less. Okay, but here only the vegetative cells will die, not the spores. When you dry, when you subject the substance to drying or sunlight, those microorganisms who have the capability to convert to the spore form, they will convert to the spore form. So only the vegetative forms are going to get killed here. Moving on to the other methods is dry heat. In dry heat, there are different methods like incineration, oven, and use of hot air oven. Now, what is why dry sterilization when compared to other methods of sterilization? Here, generally, the destruction of microorganisms happens by oxidation of cellular components. Okay, generally there are different temperatures that is applied, varying from 160 degrees centigrade for 60 minutes to other temperatures and other holding times. We will be seeing the different methods and different temperatures. You could employ, you could use your syringes, metal instruments, paper wrapped goods. All these things could be sterilized using this dry sterilization technique. Okay, the first one under that is your incineration. Under dry heat method of sterilization, that is your incineration, can be of two types. One is incineration and flaming. Now, what is incineration? Burning of the waste with minimum amount of oxygen so that there is minimum end product that is obtained. Okay, suppose you are burning a waste product in open air. There are chances that the fumes or these droplets move into the, or the carbon particle will get dispersed into the environment. Along with the carbon particle, even the microorganisms, the resistant ones, also could get dispersed into the environment. Hence, this incineration happens in a closed compartment. Okay, and with minimum amount of oxygen, so there is minimum end product or minimum ash that is obtained. Here, where do you do this incineration? Generally you find doing incinerations in hospitals, wherein you need to incinerate the bandages, the cotton swabs, all cloth materials, all these things. You because bandages are rich in microorganisms, they have pus, they have blood, all these things where microorganisms will like to survive in. Hence, this needs to be destroyed, not just anyway, but for incineration. This is the example for that. Another method of incineration is flaming. Now, here what do? Is you subject the substance directly to the flame. Where do you do this? Your inoculation loop. You show the inoculation loop directly to the flame until it is red hot, which means to say it's completely the microorganisms are completely eliminated, and then you cool it and immediately you use it. Now, here you are not getting any end product here, but the substance itself you are directly incinerating, so that flaming, so that you it's quick sterilization. You need to achieve quick sterilization. Even your forceps, you do the flaming. Okay, so this is the flaming technique. And where do? You do for spatula, you do for forceps, you do for inoculation loop. This is the example. Moving on to dry heat sterilization, another technique, that is your oven. Oven is a designed instrument wherein which works with heat energy as well as the UV. Okay, now here generally the substances which has to be sterilized is kept inside, and which is maintained for 160 degrees centigrade for 1 hour. It is electrically heated, and it is controlled thermostatically, it is controlled. Also, there is UV rays. You must have had UV oven at home, wherein you heat the food. When you heat the food, what happens generally? Why do you do that? So to increase the shelf life. So that is what happens here. Now, what are the advantages and disadvantages? It is suitable method for sterilization, so that moisture can be destroyed here. Okay, here long heating time and very high temperature, that becomes your disadvantage. You cannot do any kind of sterilization here, only the dry materials. So such kind of materials could be used. Moving on to the next kind of sterilization, that is your hot air oven. It's a modified form of your oven which you have in your hot air oven. It is an instrument which is an insulated double walled chamber. You can see it is a double walled chamber, okay, and it is insulated, inside which there is a circulating fan. There are racks kept. Why are these racks? So that you can keep your materials for sterilization. Also it is thermostatically the temperature is maintained here, wherein through the electric supply, the electric energy is converted to the so here there are coils that are placed on top as well as on the sides. There are coils that is placed. So when the heating through the coils, the heating temperature increases, and then the uniform distribution of the heat happens through the circulation of the fan. Now if you see, there is a insulated door as well, which is double walled, and also you can see this kind of material. What is this? This is a digital view wherein you can see the temperature. What is the temperature? You can also see the temperature that you have set and the current temperature. Here there are two mains. One tells that the instrument is on, another one tells what is the current temperature, I mean the whether it has reached the set temperature. There is a regulator here. This regulator helps the fan for the uniform circulation of the heat. This is what happens here. Okay, now there are various temperatures which we use for hot air oven. It again varies. There are different holding times and holding temperatures. Now what do you mean by holding time and holding temperature? This is a specific time that the temperature is held for this much time and this particular temperature. That's called as holding time and holding temperature. It's not from the time that you have switched on the instrument, but it is the time taken from the time the temperature has, that specific temperature has reached. What are the different temperatures? It can range from 30 minutes to 120 minutes, and from 60 degrees centigrade to 180 degree centigrade. So specifically, if you have to tell, it ranges from, if the holding time temperature is 170 degrees centigrade, which can be maintained for 30 minutes. If the holding temperature is 160 degrees centigrade, it can be maintained for 60 minutes, and if the holding temperature is 150 degrees centigrade, it can be held for 150 minutes. So these are the different holding temperatures and holding times. What are the items that you can sterilize here? You can sterilize all your glasswares, your petri dish, your flask, your test tubes, your powders, all these things, your metal equipments, all these things you can sterilize using your hot air oven. Now, what are the advantages and disadvantages of using this? The advantages of using this is that it is a quick process. You can sterilize most of the instruments, but then what are the disadvantages? Disadvantages is that you cannot sterilize any substance which is having moisture. For example, media is the main requirement for a microbiology lab. You cannot sterilize media here. At the same time, you cannot sterilize anything that is associated with cloth or cotton or plastic. Any of these materials you cannot sterilize here. Also, it is you need this sophisticated instrument. You cannot just use any instrument for this particular sterilization technique. What are the precautionary measures that you have to take while using your hot air oven? Precautionary measures: first of all, your holding time and temperature is very, very important. Your holding time starts only after the specific temperature has reached. It is not as soon as you switch on the instrument. And the next precaution is that make sure that there is uniform distribution of air circulating throughout, and also make sure that once the instrument is switched off, the temperature has to come down to 50 degree or below, and then only the instrument has to be opened. Otherwise, due to sudden change in the, suppose you have just switched off the instrument and the temperature inside is 160 degrees centigrade, if you open the hot air oven, due to the sudden change in the temperature, there definitely will be crack in the glassware, and also it is dangerous to the person who is operating the instrument. So this is about your dry heat method of sterilization. In the next class, we will be studying about moist heat sterilization technique. Thank you.